Joining us for our ACT-CTU clinical trial rounds. A happy new year to everyone. It's nice to see everyone back. For today's presentation, I'm honored to present Dr. Gustavo Duque, who will be talking to us about building a Geroscience clinical trial unit at the RI-MUHC. Dr. Duque is an internationally recognized geriatrician and clinician scientist whose work focuses on the biology of aging and health and age-related musculoskeletal conditions, including osteoporosis, sarcopenia, osteosarcopenia, frailty, and falls. He completed his training in internal medicine in Colombia and geriatrics medicine and a PhD at McGill University. He has held senior academic and leadership roles internationally, including director of the Musculoskeletal Aging Research Program at the University of Sydney and chair of medicine and director of the Australian Institute for Musculoskeletal Science at the University of Melbourne. He is currently full professor, the Dr. Joseph Kaufman Chair in Geriatric Medicine, director of the McGill Center of Excellence for Sustainable Health of Seniors, and principal investigator at the RI-MUHC. He also serves as editor-in-chief of the Journal of Gerontology, Biology of Sciences, and director of McGill's Division of Geriatric Medicine. Dr. Duque has led the development of multiple falls and fracture clinics and oversees an active clinical trial unit, which we'll be hearing about today, on evaluating therapies for age-related musculoskeletal disease. He has authored more than 360 peer-reviewed publications and edited several books in the fields of aging and musculoskeletal health. We are very delighted to welcome him to present to us today. Thank you so much.
Good afternoon, everyone. It's a pleasure to be here, and thank you very much, Amanda, for the nice introduction. And when I received the invitation, I was thinking, how are we going to approach this concept? Because what I'm going to do today with the audience is I'm going to start introducing terminology that I'm sure some of you are familiar with, but some of you will be really new on it. So I will be happy to answer questions at the end if there is something that remains unclear. So here are my conflicts of interest. So the first main concept I want to introduce today is the concept of geroscience. So this is a concept that started at the National Institute of Aging in the United States about in 2014, 2013, where a group of led by Professor Felipe Sierra at the NIA came out with a consideration that then was adopted and very well described by Brian Kennedy in this paper in 2014. Presented that's probably the official introduction of the term geroscience. So I will very quickly try to summarize what we understand as geroscience. So something that clinicians we experience all the time is that we treat disease by disease. So we see a patient, we identify that the patient has, I don't know, osteoporosis and osteoarthritis and diabetes, and we give them a prescription for each one of the conditions. But then the team at the NIA started considering that some of these different conditions that you see listed here, some of them actually share the same biological mechanisms. And that's why you can see on the lower part a set of what at that time they call it as the biological mechanisms of aging. And they started realizing that some of these mechanisms were activated or affected by this at the same time on several diseases, age-related diseases. So the rationale was this: if they share the mechanisms, why not to target the mechanism, and then we will treat several conditions at the same time? So we don't have to treat them separately and fragmented. We treat several of them at the same time. Not all of them. I mean, optimally it would be all of them, but that would be impossible. But at least we will have an impact on several of these conditions. And another major advance that we had in the field was the publication of this paper initially in 2014 and then updated in 2023 by Carlos Lopez Otin, an authority in the field of geroscience, and was basically summarizing the mechanism that they described and that by the NIA in 2014, and just giving a much better approach and much better understanding of what the mechanisms were. And they call it the hallmarks of aging. So what are the hallmarks of aging? Is a group of biological changes that happen in every organism that ages. But that when these changes are too dramatic or are clearly affected, then disease comes in. So and I will not go into all of them, but we have, for instance, stem cell exhaustion, telomere shortening, epigenetic changes, so mitochondrial dysfunction. So the number of hallmarks of aging went from 9 to 12, and I hope they will remain at 12. And I just want for you to keep this in mind, because that's probably the rationale, the main rationale of what do we do in geroscience to treat several age-related conditions. So the evidence of the hallmarks that are affected in age-related conditions, the evidence has increased dramatically. This is a summary of the number of papers correlate the hallmarks with age conditions. So for instance, and it's very interesting that cellular senescence has been very much associated with cancer. But at the same time, there are other mechanisms like impaired autophagy, which also has a significant number of publications linking the pathophysiology of cancer with this hallmark of aging. And you see there are many of these conditions, age-related conditions. Probably one that is missing there that I work a lot with is sarcopenia. So I would love to have also the sarcopenia included there. But at least they included osteoporosis, they included dementia, and you can see that not every condition has a significant number of publications or correlations with all the hallmarks. But there is always this connection between a specific set of hallmarks that are clearly associated with these diseases. So because we started looking at these mechanisms, and we started looking at what of these mechanisms were related. And again, I know this is more a clinical team, a clinical group, but I will start with these basics because I think it's interesting to see how we evolved from the basic sciences into the clinical trials. So there were unexpected findings. In 2020, for instance, nobody will imagine that dementia is associated with dysbiosis, with the microbiota in our guts. There was just preliminary evidence. Now, thanks to the geroscience approach, we understand much better that what we observe as changes in the brain associated with dementia, they are also triggered by the pro-inflammatory bacteria that develops in the gut of people with Alzheimer's disease. So this is something that was new, and it was through a significant number of investigators in the field of dementia and geroscience. And this table here in the right side summarizes a paper that we published recently with a fellow from Chile who summarized the evidence of which hallmarks of aging were affected in the specific case of osteosarcopenia, which is my main field of research. So people who have at the same time osteopenia, osteoporosis, and sarcopenia, muscle loss, we found the evidence from the hallmarks of aging, and surprises such as, for instance, telomere shortening doesn't play any role at all in the pathogenesis of osteosarcopenia. But other factors such as stem cell exhaustion or cellular senescence or mitochondrial dysfunction are very important in the pathogenesis of osteosarcopenia. So you see, just by considering a different approach. So let's say if we, just to give an example, and I will give you a couple of examples soon, is what if we can correct this pro-inflammatory dysbiosis in which the bacteria are in bad shape and that pro-inflammatory, what if we correct that pro-inflammatory into a non-inflammatory profile of the microbiota? Are we going to be able to treat osteosarcopenia and dementia at the same time? So that's the rationale why we are using this specific approach. So in 2019, in the United States, they established the Translational Geroscience Network for the United States. And basically saying, well, we're working on the mechanism of aging, which is what we understand now as the hallmarks of aging. And we have found so many different things. Now it's time to move into interventions and clinical trials. And they developed this specific network on a translational geroscience and we're going to talk about this soon, the interventions that will act not only on one or two conditions, but several conditions at the same time with just one intervention. And that's the goal. It's not to find a treatment, again, for cardiovascular disease or look for a treatment for diabetes. It's to look for a compound or a target or an intervention that has an impact combined into several or on several age-related conditions. And in 2023, Canada followed the initiative from the NIA, and now we have our own Canadian Translational Geroscience Network that I'm very proud to be the chair of the network. And we have members from all across Canada. We have members from the very basic sciences to the very clinical trials, from the very population studies, from the very epidemiological studies. And our major objective is to develop the translational component. We think that for the critical mass of people that we have in Canada, we have the potential to become an important engine of in terms of epidemiology and translational translation into clinical trials and also evaluation and community settings. But I have introduced you with the two concepts so far, the geroscience, the hallmarks of aging. And this is a concept that is very important for the understanding of what do we do in terms of geroscience and in the geroscience interventions and gerotherapeutics. We have the lifespan, and I'm sure most of you understand this very well. We have our lifespan, which in humans, it will go up to 20 according to some theories. So basically we have, that's what we can live. But then there is the health span, which is how much or how long we can live in absence of this capacity of disease that affects our function. Yeah. So if we have a non-optimized health span, we will start having age-related conditions always arrive. Sooner or later, we are going to have at least 2 or 3 age-related conditions, either diabetes or osteoarthritis or osteoporosis or sarcopenia or any of them, or several of them. But the idea is that those diseases can affect very early on the way we function. So by treating them, by studying the interventions, by doing some prevention, we're going to be able to prolong and optimize the health span. So health span in geriatrics and in geroscience is one of the most important outcomes. So as long as the other conditions and all the other medical conditions are controlled, then we can assure that the patient will have good health span. And this is important to highlight this now because some groups are promoting what they call anti-longevity clinics or anti-longevity promising people that they are going to live until 120. This is not what we are in, what geroscience, what the principle of geroscience is. We are not looking at extending the human lifespan. We are aiming to extend and optimize the human health span and reduce the amount of time our older persons experience with disability and with limitations, severe limitations in activities of daily living, basically. So, and they maintain their independence. I think that's important because I'm going to mention that later on several times. So the is moving very quickly. And I will just give you a couple of examples of what we are doing at our labs. Again, I know that the interest of this group is more clinical, but this is the rationale of how we end up doing clinical trials on geroscience. So there are some priorities at this moment that we are working on. We're working on what they call senescence-associated secretory phenotype. This every organ that is affected by a disease or an age-related condition has a higher number of what we call senescent cells. So those are cells that are even, within every organ, within our liver. We get older, we start developing these senescent cells. And these senescent cells are important in the sense that they secrete pro-inflammatory factors. And it's what we call the senescence-associated secretory phenotype. So it's very bad if our brain is full of senescent cells, of course. But they are also a mechanism of protection. So, we cannot inhibit completely. And that gives to what I'm going to mention later on, one type of gerotherapeutic that is called senolytic. What does it mean? That goes and kills the senescent cells within the target organs. So we can use these senolytics and give it to the patients and target the specific organs, and that's what we're working on. We can also look at metabolomics and lipidomics. This is mostly to develop biomarkers, and we have been able to develop new animal models to study aging and geroscience. I will bring you two examples of the preclinical, where we're reaching now. We finished the preclinical evidence, and now we are moving into negotiations and with commercialization and strategies and all this to develop the compound, finalizing the evidence to start aiming to start the clinical trials in humans soon. So I wrote two quick examples. One of them is something that we observe at the same time in muscle and bone, and from the area that I work and my lab. So we observe that there's a lot of fat in the sarcopenic muscle inside the fiber, around the fiber, and it's very similar to what we observe in the osteoporotic bone. We see a lot of fat around the bone marrow, even in the lacunae, and we know that these two fats, which is interesting, that's something that we did, these two types of fat, the marrow fat and the muscle fat, are very different than the other types of fat. So we did a complete lipidomic analysis of those fats, and we were able to report that muscle and bone fat share 92% of similarity in terms of the composition. And they are different than the subcutaneous and visceral fat. So it's not obesity. It's not a story of obesity. This is a story of an alteration on the hallmarks of aging that triggers the development of fat in these two tissues. So the other thing that we found very interesting is that this fat is more inflammatory, very inflammatory, like inflammation. I probably have captured. I have mentioned inflammation several times because it's very important hallmark of aging, and they are more toxic. They are lipotoxic. They secrete factors that are relatively similar to the factors that are secreted by senescent cells. So we have all these different factors that are secreted and triggering all these mechanisms, these hallmarks of aging. We have compared very, very extensively fat from the bone marrow and fat from the muscle. And without going into all the details, we know that they are induce very significant catabolism and affect the growth of those tissues. So the rationale to develop this from the geroscience perspective was, okay, if those fats are so similar and they secrete these toxic factors, what if we inhibit the capacity of these fats to secrete those factors? Are we going to regain not only muscle, but also bone? And this is the evidence. We block it with an inhibitor of fatty acid synthase. So that fat has enabled to produce fatty acids. So the toxic elements are not secreted. Again, going very quickly, we treated mice, osteosarcopenic mice, and we were to them from osteopenia. And we were also able to revert their sarcopenia, that they gain muscle and bone. So the bottom line of this approach, again, not going into all the details, is that if we target fat, this specific type of fat, we're going to gain muscle and bone at the same time. Are there other benefits? We don't know yet. I mean, we don't know if it's a cognitive or cardiovascular or cardiometabolic benefits. Definitely, their cardiometabolic deficits will improve because this inhibition was not specific necessarily for those types of fat. It inhibits all the other fat, but this is what we're working on. And quickly, the second one that I mentioned before about the dysbiosis and microbiota. So my team has been working in the last 6 years on a specific pathway of tryptophan degradation. So, you know, tryptophan is an essential amino acid, and it can be metabolized via the pathways of the serotonin, which is probably the product that is the better well-known of this pathway. But most importantly, 95% of the tryptophan is metabolized by kynurenine, by the pathway. Why I'm telling all the story is because when we have, and this is something that we know now, this pathway is involved in GI function, is involved in mood, is involved in cognition, and is involved, and this circle is the one that we generated from my team, muscle and bone. How does it work? So again, hallmark of aging. Dysbiosis is a hallmark of aging. Abnormal bacteria, a normal microbiome generates an inflammatory milieu. Here is the pathway, the kynurenine pathway. And you see that there are some in red and some in green. The ones in red are anabolics. They stimulate or facilitate bone muscle formation. They are neurotrophic. The one in red are toxic, are pro-aging. And what happens when we have normal bacteria and microbiome? The tryptophan is metabolized all downstream to this compound, picolinic acid and NAD+. If we have an abnormal bacteria, people with osteosarcopenia, osteoporosis and sarcopenia, they have dysbiosis. We know that very well. They don't produce this. They stop here. And what happens when we have too much of that, specifically too much of kynurenine, and we have already done it in humans as well, then it triggers hallmarks of aging, oxidative stress, marrow adiposity, autophagy, all the other hallmarks of aging, and triggers inflammation. So again, I'm sorry if I'm going into too much biology here, but it's because this is the rationale. So what would be the rationale here to use geroscience in this kind of preclinical study? Well, we have two choices. We have three choices, actually. We have several choices, but we selected three. What about if we normalize the microbiota? Or what about if we inhibit the production of kynurenine? Or what about if we give picolinic acid? So we selected this one. And as in the previous experiments, we gave picolinic acid to mice, and we recover them from osteopenia, osteoporosis, and we also them from sarcopenia. And even we went beyond that. We did experiments with Jeremy Van Kerkhove here at McGill, and we treated C. elegans, which is a model, very interesting model that we used a lot for aging and geroscience. And we also treated zebrafish. What we found is when we treated those with picolinic acid, they were able to, in the case of zebrafish, they swim more. And this is an indicator of health span. Yes, we have been able to reduce inflammation. This is a very good question because we did it through the SASP, the secretome, the inflammatory SASP. So by using arrays, we did that. So here is where we are, and here is it means that we are ready to use picolinic acid and translate picolinic acid into phase 1 trial. And picolinic acid can be medicalized, like make a drug. I don't know how do they say that. Drug sites. Okay, druggable, make druggable, and same with the other approach of inhibiting the fat. And some people are actually using these GLP-1 agonists, trying to demonstrate the same. Moving forward into something that you are more interested on. We are also doing translational research in terms of using longitudinal studies, longitudinal cohorts, to evaluate potential biomarkers. That could be biomarkers for several conditions, predictors of several conditions. Again, from the geroscience perspective, we don't focus just in one, we focus on several, as many as we can. So we had access, we obtained access to the CLSA, to the Canadian Longitudinal Study on Aging, the MUHC, and the Cartagine. The three of them have Canadian cohorts. And now we're evaluating this for the use of geroscience. But the main objective of my talk today, after giving you the whole rationale of where do we come from, is that we create the first in Canada geroscience clinical trials unit here at the Research Institute. And I would like to thank Dr. Angela for the support to this clinical trial unit, for giving really a very active support and assistance to the initiation of our geroscience clinical trials. So what do we want? What is the objective of this unit? Is to move away for the reactive model, where we only treat disease by disease. When the patient comes with a glucose of very, very high, we give them something and we push that glucose down. High blood pressure, pressure down. So in most of the cases, those patients will end up with the multimorbidity of polypharmacy. That's what I see in geriatrics all the time. So we are moving into targeting aging. I don't think I have to go onto this again. Targeting the hallmarks of aging, to go into mechanistic interventions. Mechanistic interventions that may have an impact on several conditions at the same time. And then other preventive type of interventions that also improve health span. The health span is not only with a tablet. I don't think we're going to reach a tablet that is not alone. You know, we have to combine it with exercise, with leisure activities, with social activities, with nutrition. But that's the principle. We are trying to move away from that very much reactive model of care. But we have problems. We have challenges, not problems, but challenges. And this has been recently summarized by an international consortium on geroscience, on gerotherapeutics. So the other concept I want to share with you today is the concept of gerotherapeutic. So gerotherapeutic is defined as an intervention, pharmacological or non-pharmacological, that has an effect on several hallmarks of aging. and therefore will have a beneficial effect on several age-related conditions. That's the definition of gerotherapeutics. Without going into all this, we found that one of the big challenges is that some people, the big discussion is whether aging is a disease. If you ask me, my personal opinion is no, it's not a disease. I mean, it's a process, it's a biological process, but it's not a disease. It triggers in the presence of risk factors, it triggers disease. So we have to target the mechanisms that are triggered with aging in order to prevent or treat those conditions. And it's very difficult also because at this moment, if we apply for to the FDA for a drug or anything or intervention, then they will come back and say, what is your outcome? What outcome? Is aging? So are you going to do a clinical trial with 200,000 people and see if they die younger or older with that drug? Or the other thing, and this is something that I will mention later on, is the industry. But we find probably the main barrier is in the policy and in the actually inside the medical field. Because it triggers also some kind of prevention for people saying, oh, so then I will not have the monopoly of treating just coronary artery disease or hypertension. Yeah, so there are also some kind of political issues that have been triggered, but there is definitely a discussion, a good discussion now of what will be the health-related endpoint in gerotherapeutics and geroscience. So some people say, what about total mortality? But it will be hard to really document the reason of death. And again, the sample size should be very, very high. The disability-free survival, which could be correlating to health span. I think this is something that is easy to measure in terms, especially the recovery. If they lose health span, then we can, show that they have better mobility. In terms of multimorbidity, are we going to document all the different chronic conditions? So it becomes also more like the old model, like a reactive model. And then it comes to frailty, which is a concept that we tried for the last 30 years in geriatrics to operationalize, and we never operationalized it. And never was implemented widely in clinical practice. It's only very narrowly identified by geriatricians and some of our colleagues who like geriatrics or like seeing older persons, but it's not a concept that has been operationalized. This is our main issue at this moment, is what are the outcomes that we are going to propose in terms of what will be the therapeutic impact of the gerotherapeutics. So now we have a new concept, and this is probably the sixth concept that I'm going to share with you today to introduce, is the concept of intrinsic capacity. I don't know if you're familiar with this concept, but this is a relatively new. It was adopted by the World Health Organization and basically has 5 dimensions: cognition, locomotion, sensory, vitality, and psychological. And we measure this in a way that we can come out with a construct or a way to measure it. And there are several constructs. This is one of many in which we have each one of the domains is measured, and then it comes out with a score, and we can tell the patient, your intrinsic capacity is 30% of 70%. If you have a 70% intrinsic capacity, the probability that your health span will be affected is low. If you have 30% intrinsic capacity, you're probably in troubles. If you have 10% intrinsic capacity, you are probably in a nursing home, or you are really, really disabled. So it's more or less the way we are going toward at this moment in the field. We are going away, moving away from frailty. We are moving away from other variables. And we think that at this moment, the components of intrinsic capacity are the ones that we are going to be able to measure. However, and despite, frailty never had an international code disease, never. And intrinsic capacity already has one, and it's this one, aging-associated decline in intrinsic capacity. So this is our justification for the FDA, and we can go and say, we have the code, we have how to measure it, and now we have to treat these patients with gerotherapeutics and see what the response is. Again, but this is always a however. Yeah, Susan.
Sorry, I'm just looking at this. And so the question I have is how sensitive is this to change in terms of, chronological age goes on. And, if you already are at the total severe loss or, kind of at the extremes of your loss of intrinsic capacity, can this be sensitively reversed, with treatment?
That's exactly the question that we are trying to answer, because, for instance, sensory, just to give you an example, sensory is a dimension that is very easy to correct. With the hearing aids and a pair of glasses or a surgery for cataracts, we try to correct that. There are some others, like locomotion, but we definitely can look at we have the instruments to measure them. And some of these instruments are very sensitive. We're not going to all of them, but for instance, the SPPB, the short physical performance battery, is very sensitive. So, but coming back to your question, yes, and that's why we have two challenges from this concept. The first one is how we are going to unify the way we measure intrinsic capacity, yeah, as an outcome. And second, as you clearly say, is, how are we going to measure improvement? What element of this will have the most important impact on improvement? Yeah, I'm biased, completely biased, but I think it's the musculoskeletal component, because musculoskeletal component is connected to all the others. But I'm biased because that's what I do every day. So to answer that question, we established a CIHR-funded project that is called the Thrive project, which started with a few data sets, and now we have 15 data sets in 10 countries. It's a federated research. It's not that we're not going to collect a big data set. We're just going to analyze at each one locally. And what they are doing at this moment with the Thrive project is, including the CLSA here in Canada and MUHC, is we are going to develop a construct for intrinsic capacity with what they have. Yeah? So we don't want to narrow the concept into 5 criteria, like Fried's criteria for frailty. We don't want to create that because it's not a flexible construct, a flexible model. So what we are doing now with the Thrive project is developing the construct. So we'll have probably 15 different constructs. But at the same time, we're going to use those constructs and look at the predictive value for adverse outcomes. And every cohort has more or less adverse outcomes. Yeah. So with that, we are going also to start playing with in some sense playing with the components and removing one and re-performing the analysis. And this is what we're doing at this moment. So the idea is that we can remove, and then we only need to do a grip strength and a DEXA scan, and that will be enough, which will be great, which will be enough, or just a MOCA and a vertebral calcification, aortic calcification, sorry. Yeah, so this is where we are at this moment. Again, we have now quite a significant, from all the continents, all races. We have Chinese, we have Japanese, we have Australian, European, South American, and Canadians. Okay, last part of my talk, moving into clinical trials. So we are working on the concept, but we need also to start, it's already started, the clinical trials. This is a relatively recent summary of some of the drugs that were that have been tested as gerotherapeutics. I will not go into all of them, but you can see, for instance, that some of them are related to diabetes treatment, and there was a big interest on the metformin study that unfortunately didn't work well. So this is just an example. So basically what we do here is coming from the preclinical, like the things that I did, like I show you today, and then looking at the hallmarks, testing that in preclinical, and then moving into humans. This is where the intention is. And this is an example, for instance, this is a study using a gerotherapeutic, a senolytic, so a drug two drugs combined that kill senescent cells. This is an osteoporotic woman, postmenopausal woman, by Sandeep Khosla from the Mayo Clinic. And they didn't find a dramatic effect, but there was an improvement in terms of bone mineral density in the woman treated with this serotherapeutic. What they didn't do is look at other organs. So they didn't look at muscle, they didn't look at other things. So they're trying to solve that problem with a different one. This is another clinical trial we're participating. It's an NIA funded. Initially was by the Buck Institute in California, Ohio State, University of Connecticut, and now McGill. We are bringing this to McGill, in which we are going to look at the 180 older persons. We are going to look at the hallmarks. So it will be more focused on the hallmarks. And I just would like to share with you probably the major accomplishment of our teams here. I don't know if you're familiar with the X-Prize health span. This is a model, very interesting. It's a model in which they give you the money. It's not a grant, it's a prize. So they give you the money for you and recognition of what you have done already, which is good. So 740 groups participated worldwide, and for the first phase, and they selected 40, and they gave some money, $350,000 to those 40 groups. And we were the only group in Canada to get it. So, and when I say we, I'm talking about the Translational Geroscience Network. So just to give you an example, look at what they want. And they say you must show improvement in 2 out of 3 measures of these components. And we have to use the tools that they request. Doesn't matter what intervention you want to give them. You can give them just exercise or vitamin D or I don't know, but you have to measure this in healthy older persons. That's the challenge, is to find the healthy older person. But the healthy older person is persons who may have comorbidities. It's very hard to find an older person without a comorbidity, but who are controlled. Anyway, just to give you an example. So there are, so if we treat them, and it has to be, the response has to be within a year. Yeah, that's short, especially if you are going to measure cognition. I mean, cognition, you see that the big cognition studies start working about two years, but if they work. But it's challenging. What is the second phase? The semifinals. They are going to give 10 awards, 10 million, to complete the clinical trial. So the initial one is to start recruitment and the pilot and feasibility, and the second one is to actually start the clinical trial. This is due this year. And then from those 10, they will select the final, will be the compound or intervention that has an impact, significant impact on this criteria. That will be $60 million. Okay, so that's I wanted to share this with you because this is where we are moving toward in terms of we are very clearly selecting the variables that are associated with the hallmarks of aging. and with intrinsic capacity.
So I don't see anything for inflammation, or am I wrong?
In this one, is just the not the inflammatory, but that's the other thing. The groups can add things, so we added a much wider inflammatory profile. Right? Yeah. So at this moment, this is our evolution proposal. We are giving our patients, our participants, and it's starting soon here at the Research Institute. We're starting soon with HMB, hydroxymethylbutyrate, which is a metabolite of leucine with wide biological impact that has been demonstrated to improve mobility in muscle in people with sarcopenia, but has also recent evidence showing that it may have an impact on cognition as well. And we are going to combine it like in a randomized controlled trial, combined or separately with this 2-HOBA, which is a compound from natural origin, but has a very significant impact on inflammatory biomarkers. And that's why we are going to avoid the inflammatory profile. But very interestingly, has also recent evidence has an impact on cognition. So we expect that by combining these two compounds, we will have a combined effect on multiple hallmarks. So we're going to correct inflammation, autophagy, proteostasis, maybe microbiota, who knows? But that's the goal. And this is Guy Boutros, who some people already know at the center, who is the postdoc now in charge of this protocol. And this is another one that we are doing in Australia with Ben Kirk, another of my postdocs, looking at the effect of HMB and vitamin D in people with osteosarcopenia, but we are also targeting several of these hallmarks, like similar way of doing it. So in conclusion, the way I understand it, the way I see it, is that Geroscience has revolutionized the way we understand, identify, and treat age-related conditions. Our way of thinking has changed completely in geriatrics and Geroscience. Intrinsic capacity is the most important outcome, I think, when associated to health span of gerotherapeutics. The challenge, again, is that we need a unified way or a wide way of measuring it in an effective manner. The number of Geroscience-based clinical trials and drug companies is increasing. This is just an example of all the drug companies who are specialized in Geroscience. Some of them are repurposing drugs. Some of them are creating their own compounds. All of them are interested in coming to Canada. And we are in the process of negotiating, because now with different environment in the United States, now they feel that they are very much welcome here. So our strategy for the Research Institute and for the Clinical Trials Unit is to bring as many of them as possible. And we already have 3 of them engaged. To one of them who is a co-sponsor of the clinical trial, the one with HOBA and HMB, and 2 other companies. And now that there are so many, interestingly, Pfizer is reopening their Geroscience unit. And Novartis is also starting to open the Geroscience unit. So that's where the opportunity is. I think that's where we are going to really find the niche for geriatrics, Geroscience. And this is not unique for geriatricians. And this is my last comment, because I know in the audience there are specialists in many other fields. This is not unique or exclusive to geriatricians. This is for everybody who deals with multimorbidities in your older patients. You know, this is cardiometabolic. This is, if you are a cardiologist who is running a trial on maybe we can add the cognition component. So as long the criteria that we have for a Geroscience trial is that it should target at least 2 out of the 5 dimensions. Yeah, so dimension and depression, osteosarcopenia and cognition. I can give you tons of examples. And also for the people interested in cancer, which is a major strength at our research institute, some of these chemotherapies have demonstrated to be senolytic. they target senescent cells, and they are being tested for senescent cells. So we can use the drugs that you are being so familiar with for so many years. We can test it if there is a possibility of testing them for age-related conditions. So with this, I would like to thank my team and all the people who pay the bill and all the people who collaborate with us from multiple places, from the different perspectives, epidemiology, basic sciences, but all around the Geroscience. And this year, the conference of the Translational Geroscience Network is in Vancouver in November. So we will circulate the information soon. Everybody is welcome to attend and participate. Thank you very much for your attention.
Hi, I'm Tania Ferreira. We've met before.
So
thank you so much for this very exciting presentation, an excellent presentation. So I'm wondering in terms of the gerotherapeutics, so where do you think the field is going into more the pharmacological or non-pharmacological interventions? Because lifestyle interventions have been there for a while now.
Yeah.
And we know they work for so many different outcomes. And we know that everybody should sleep well, eat well, do exercises. Is there anything very new in this field in terms of the non-pharmacological interventions that you think this field should be looking at compared to the non-pharmacological, to the pharmacological one? What is your...
I think the answer will be both. And regarding the non-pharmacological, probably the one that has more future is the nutraceuticals and nutritional interventions. And the new nutraceuticals, something like we're doing with the picolinic acid, with the HMB. So instead of giving just nutrition, like, more food or more amino acids, we just try to identify the downstream, which metabolite is the strongest one. So for instance, with HMB, we know that it's 30-fold stronger than leucine. So we can, that's why and with picolinic acid, we can also, so that will be probably where the nutraceuticals and the non-pharmacological, because I agree with you, well, exercise is good for everything. So we just recommend, but exercise is good, but it's not able to actually revert to, or treat the specific set of comorbidities that are very common in older persons. The other one, and then the pharmacological, all those pharmaceuticals are interested in pharmacological, of course, and developing their own compounds. The question sometimes is how much, as we did with the osteoporosis field, we combine the treatment with vitamin D and calcium. So how much are we going to play with those compounds in terms of combining it with other non-pharmacological nutritional interventions or exercise and something else? But some of these compounds are, I didn't mention, for instance, this one of them, Longeveron, is a company in Miami who is giving, because one of the hallmarks is stem cell depletion. So they treated frail older persons with the infusion of allogenic stem cells, mesenchymal stem cells, and they found response. So it's not only the actual compounds, but also interventions like the stem cell, but the serious ones, not the ones that they are, in who knows where they're injecting those things, because that's the other thing. There's a group that I mentioned that is taking it into anti-longevity, which I think is not as scientific and evidence-based as was this field.
Hi. So, so there's a lot of talk also about the, intermittent fasting may be helpful as well in trying, in this. And so I guess my question is, there's all these abilities to measure biological age, epigenetic clocks and all these clocks. And so the question I have is, are these markers, like you measure them cross-sectionally, and then you have your biological age? And do these change over time? Are you able to actually modify them enough that you can actually say people are, or reverting in terms of aging? And because when you look at this, at these interventions that you're describing, these seem actually to me much more targeted to certain pathways and everything. Exercise, intermittent fasting, all these things are kind of more like a general approach. And so my concern is also that there are multiple things out there, as you say, who are trying to improve longevity via various ways, and whether this could, be actually more detrimental to the field than anything else.
Yeah, it's true. Well, I'm glad that you mentioned about the epigenetic clocks, because they are becoming less and less popular. And the reason for that is because they are a good indicator of our epigenetic situation. Acetylation, histone acetylation. That's only that. A picture of how much acetylation you are doing in your histones, or whatever clock you are using. But they are not really. They are not in the global. That's why intrinsic capacity, I think, has the potential, because that way, and maybe, because we don't know yet, maybe, and that's part of the TRI project, is maybe we'll be able to identify biomarkers that correlate very well with intrinsic capacity in a way that you want to see improvement, then you follow those intrinsic. But definitely it's much easier to bring somebody from intrinsic capacity 40% to 60%, and you can have it all like measured, than just one element, one hallmark. I mean, those epigenetic clocks is one hallmark out of 12. So you are only evaluating. Again, it's not very popular. It's decreasing in popularity, but some longevity clinics, they do like a checkup, executive checkup, and a clock, and they say your age is this or that. I mean, this is not necessarily correlate very well, but intrinsic capacity, you can tell, okay, you have a very bad shape. We have to target that. Yeah, it still is useful especially in research, but I don't think in the translation into practice is going to be, or in geroscience use.
Yes, I think I was told to go ahead. That's very interesting, Dr. Duque. It's a bit maybe a broad question based on your vast area of research, but are you trying to increase the lifespan or make the health span closer to the lifespan?
I think it's mostly to increase the health span. I think so to prolong the health span. I'm not convinced that we are going to be able to extend the lifespan too much I mean, it will be probably an additional gain, but probably the most important objective at this moment is health span.
And in your measure, I guess it's not so much a measure, but an exposure, but what do you think about blue space and aging?
Blue space is
okay.
Sorry, I'm not familiar with
it's okay. Thank you.
Okay, we have one local and probably the last one, or?
Yeah, thank you. That was very interesting, and I learned a lot about clinical trial units. And if I got it right, you're going to follow up these patients for 1 year?
Yes.
I'm just curious about what kind of retention strategies you have, or do you expect a lot of loss to follow up, or how are you going to deal with that? Thank you.
In terms of retention, the thing, because these patients are considered as healthy older volunteers. As you know, the likelihood of people participating in clinical trials, if they are healthier, is much higher, because these are volunteers that do it because they are motivated by their nobody specific condition. So that's we expect that the retention will be much higher than the regular retention in people with the chronic conditions. Okay, thank you very much.
So thank you everyone for joining us, and thank you so much, Dr. Duque. And we also welcome you. We have a clinical trial symposium to hear other amazing work at the RI. But thank you so much, and see you next time.